The Indian Institute of Technology Bombay's Biosciences and Bioengineering or BSBE department is home to high-end microscopy and lithography facilities that researchers can use to image tissues, cells and organelles, measure physical and electrical properties of cells, and print micro-sized objects. The BSBE department allows affordable access to these facilities to researchers outside IIT Bombay. A laser scanning confocal microscope uses a laser source and pinhole to block out-of-focus light from the sample. Using optical sectioning to capture images of the sample in different planes and reconstructing these using software, it can create three-dimensional images of samples. The laser scanning confocal microscope at IIT Bombay can make high-resolution images of various types of samples such as mammalian cells, bacterial samples, proteins, yeast samples, and parasites. It offers a temperature and carbon dioxide controlled incubation stage for long-term live cell imaging and non-descanned detector for use with the multiphoton laser for deep tissue imaging. A spinning disc microscope is a confocal microscope with a disc with several perforations spinning at a specified speed that helps illuminate the entire sample area at one go. Spinning disc confocal microscope facility at IIT Bombay is a computer-controlled fluorescence microscope fitted with high-speed microlens enhanced NIPCO spinning disc. It is suitable for capturing fast-changing phenomena. For example, it can capture swimming bacteria at 50 frames per second at full resolution. It is a very powerful tool for live cell imaging over hours to days. It is suitable for imaging fast dynamic processes and multi-position imaging. The 3i lattice light sheet is a state-of-the-art machine that can give high-resolution images at a high speed without bleaching or degrading the samples. It can capture 200 planes per second, and one can image samples for days and weeks continuously without any degradation. A spatial light modulator shapes the laser beam to create a light pattern. The illumination and detection objectives are designed to give better contrast. Equipped with a temperature and carbon dioxide control chamber, it is suitable to image live cells. The atomic force microscope or AFM can be used for topographical imaging of samples. The AFM at BSBE can handle soft and wet samples, that is, biological cells and tissues, and hence it is called a bio-AFM. One can measure topography, morphology, and mechanical and electrical properties of cells and tissues. Pre-processing of samples is not required while using the bio-AFM, and hence it can be used to study live cells. The versatile bio-AFM can be used to measure physical properties of cells such as its stiffness and conduct single-cell experiments. The bio-AFM at IIT Bombay offers contact and tapping modes of operation for imaging, force mode of operation for measuring mechanical properties of samples, conductive AFM, piezo force microscopy, and nano indentation. The facility is used by the BSBE as well as other departments in IIT Bombay. The fluorescence activated cell sorter at IIT Bombay can be used for applications such as cell apoptosis analysis, cell proliferation assay and cell cycle analysis, protein engineering, and isolation of immune cells for immunotherapy and immunophenotyping studies, etc. The detectors are designed in an octagonal pathway in order to minimize signal loss. The machine has a Class II biosafety cabinet integrated with high-energy particulate air or HEPA filters to avoid aerosol contamination. It can detect samples with size ranging from 0.2 to 25 microns. However, the team has been able to detect samples smaller and larger than the specified range. The 3D laser lithography facility at IIT Bombay can be used to print microscale and nanoscale 3-dimensional and 2-dimensional structures. One can fabricate microscale structures on ITO-coated glass and silicon wafers and nanoscale structures on borosilicate glass cover slips and fused silica glass substrates. Due to its speed, precision and resolution, this 3D laser lithography system enables the experimentation possibilities in many research fields such as microrobotics, photonics, electrochemistry, microfluidics, tissue engineering, and electronics and material sciences. It has been used for printing microneedles and microfluidic channels. A full list of facilities is available on the webpage of BSBE department. Please feel free to contact us if our facilities can be used for any of your research requirement.